DSV is a venture builder that, unlike traditional VCs, builds startups from the ground up. So they bring together entrepreneurs, scientists, and create teams to tackle specific challenges. Laura is head of partnerships and venture portfolio pharma at DSV, and she brings a deep expertise in biotech with a strong focus on IP, tech transfer, deal making, and fundraising. And Kerstin is director pharma at DSV with over a decade of experience in oncology, biotech, and pharma sector, with valuable insights into translating cutting edge science into impact. So welcome to both of you. We'll have time for questions at the end and introductions. And we'll be recording this first part of the webinar so that we can share it with others afterwards. So over to you both, Kerstin and Laura.
Fantastic. Thanks, Jane. So I guess no need to introduce myself because Jane just did such a lovely job of that. Just to say that before being at DSV, I spent a lot of time in the charitable sector myself as well. So I spent more than 10 years working for the commercial arm at Cancer Research UK, Cancer Research Horizons as it's now known. So I'm very familiar with your world and all of the efforts that non-profit organisations are making to drive patient benefit through innovation. And I guess what we want to tell you about today is how Deep Science Ventures goes about creating impact in our own way, and maybe just to sort of challenge the thinking a little bit and put to you really that invention is actually the key bottleneck that we need to solve to unlock innovation moving forward. And I'll sort of tell you a bit more about our thinking there and then how we go about invention at DSV, and Kerstin will take you through some details and sort of real life examples of how we've gone about doing that of late. So just to kick off then, invention versus innovation. I don't think it's something we very often talk about. What is the difference here? So the kind of simplest way to look at it, I guess, is that invention creates something entirely new. So you might think about something like the invention of the light bulb. Innovation then improves upon existing creations, finding new applications, incremental improvements to those inventions that went before. So invention of light bulbs then drove, vast changes, for example, in working practices. People could now work in outside of daylight hours. It drove power grids, power stations, so many different evolutions then of what you can do with that technology. But the invention of the light bulb was the foundation that underpinned all of that moving forward. And you could look at also at examples like the evolution of a phone into, the little computers that we all now walk around with in our pocket. But without the invention of telephony in the first place, none of that innovation could have happened on top of that. So invention really is something completely original, so a new concept that did not previously exist. It tends to result from highly creative thinking, which of course builds on the knowledge base that is in existence, but puts the pieces of knowledge together in a new way that nobody else has yet managed to do. And very often inventors are specifically looking to solve challenges, and they are inventing with the explicit purpose of solving a particular challenge or a need in society. So the process, just to break it down into really simple steps, where you would start here is actually what is the need that we need to address. Then there's a period of ideation. So what potential solutions to that need could be created that don't yet exist. There's an iterative process of building prototypes, testing, improving upon that, until ultimately you have an invention that can help you to address that particular need. So the principle here that I'd like to put forward is that invention is actually the foundation, the bedrock on which innovation can be built. Ultimately, innovations then can be developed into implementable technologies to achieve all of the patient benefit goals that we're all caring about in our roles day to day. One thing that I thought was really interesting in terms of, do people think about invention as opposed to innovation? I just plugged the two words into Google's database of words. I don't know if you've ever tried this, but it's quite cool because you can just kind of see over the years, what are people actually talking about? And you can see here since sort of 1990, and this is, really familiar with my own career path, actually, the emergence of innovation as a growing concept. And this is probably around the time also when lots of universities became professional in their tech transfer services, lots of charities built tech transfer teams and started funding innovation grants. If you look at invention, it's pretty much flatlined. People aren't talking about invention in the same way they're talking about innovation. So we see intentional invention as a sort of a neglected component of technological progress, which obviously, looking at this pyramid, if you can expand that base of inventions, you can then proportionally expand innovation and the resulting technologies. If we're stagnant here in invention, then we are obviously rate limited in terms of innovation. So where DSV is focused is in the foundation here, in generating new inventions to address specific needs that don't yet exist. So just to recap, the differences here between invention and innovation. So invention is entirely new concepts. Innovation is improving upon things that already exist. Invention has the potential to be disruptive, perhaps groundbreaking. The light bulb example is one to keep in mind there. Innovation is more around continuous evolution and improvement. Invention moves us possibly into completely uncharted territory. So we see even amongst our portfolio companies that we build that very often they're taking quite disruptive approaches compared to other companies in the space. So that comes with sometimes higher risk, but also the possibility for much higher impact. Whereas innovation is possibly a more calculated risk-taking approach because it is building on the knowledge of what went before. Obviously, the time taken to move from the very first invention can be longer because you're starting really with an entirely new concept. But very often, as Kerstin will sort of tell you a bit more about, we take a sort of engineering style approach to this. So perhaps the different building blocks that constitute an invention themselves have been somewhat tested and de-risked. So we do our best to speed up that pathway. So yeah, then we're going to spend a bit of time just telling you about how does DSV work in the invention space. So I was just going to give you a very, very brief introduction here to DSV, and then Kerstin will take over to try and bring to life a bit what does invention look like for us day to day. I know before I joined DSV, it was very hard to kind of picture how does this actually work? How do you actually go about moving from a problem to a new invention? So what does DSV do? So we are a venture builder. As Jane eloquently explained at the start, we bring together all the different components that you would need to launch new companies. So we use scientific knowledge that already exists to create potential new solutions. And we bring also into that founders with what we believe to be founder type entrepreneurial characteristics. And we nucleate investment networks around that to launch those companies so that they can then undertake the proof of concept for the inventions they've come up with. Kerstin and I both sit in the pharma team, but DSV actually acts more broadly. So we work not just in therapeutics, but we also work across climate and agri-tech and also the computation sector as well. So Kerstin, I was going to hand over to you at this point. Probably best if I keep control of the slides and then just flick through as move through.
Cool. Super. So this is walking you through how on earth we even come up with these novel inventions, which is actually a very structured process. So it actually starts from the beginning with defining an outcome that we want to achieve. And it's really important that you set this based on the state of the art that we find in a specific field, but also as ambitious as you possibly can. I often like to use this kind of analogy of if you're basically trying to fly to the moon, then you need to be able to design something that has the chance that gets there. You won't find anything, you won't be able to get to the moon if you're just designing something that gets you to maybe New York and then bolt on some something that's disease modifying, managing, depending on what's out there. Once we have that outcome, then we basically determine some requirements. So this is like the space that a solution must operate within. So it could be like in the cancer space, for example, you need to have a solution that has efficacy as a single agent because pharma companies won't buy anything that is just active in combination, something like that. So we try and garner as much requirements as we possibly can at the beginning. And then from that set of requirements, we then narrow down to sub outcomes that we think we can actually achieve. So that could be a cure in patient population X, or that could be a disease modifying solution in that X in a certain manner, for example. And once we have that opportunity area, that then allows us to really think about what is actually holding back the field, what have people tried and why hasn't it worked and why have they failed, to then understand what has gotten in the way and what could be done better to resolve these constraints. So that then leads to the generation of approaches that you can then develop further into actual innovation and invention. And that you can then use as the basis to really form your company that you're looking to spin out in the end. And I understand that this sounds very, very highly abstract. So the best way to actually show off how that works is to walk you through an example. And we've chosen an example where we have worked with a charity partner, the Children's Tumor Foundation, which is a charity partner focused on NF, which I'll walk you through in a minute. However, a lot of the time, people are asking, how do you even do this? Where do you find all the information that allows you to do such a process? So we have basically developed our in-house tools that actually allow us to do that process very, very quickly, right? So we have generated an AI that's an LLM system, which is called Elman, that basically supports all of our scientific ideation diligence in order to create our inventions that will actually hold true. So all of the things that we would usually do in the process of creating a new company, starting out with the ideation phase, are supplemented by Elman. So you could basically ask Elman to give you the state in the art in a current field, and 5 minutes later, you have a really, really good view of what's currently happening in the clinic and why things are failing. If you have a solution that you think has legs, you can ask it to do a health economic analysis to understand whether your solution falls within something that's actually acceptable or whether you need to go and change it. You could also ask it to do like a pattern analysis in a certain space to understand if you have freedom to operate and whether there's space to operate, or whether what you are doing is not novel enough and you need to shift your proposition. And that basically allows us to go from an outcome that we're wanting to achieve to something that we can invest in the space of 12 months. Super. And I think now we're launching into this example that I've just mentioned. So this is a collaboration in neurofibromatosis with the Children's Tumor Foundation, which is a charity that's solely dedicated to solving NF. So what the heck is NF? NF is actually the most common rare disease that is out there. It's actually 3 times more prevalent than cystic fibrosis, which I'm sure everybody here on the call has heard of. and it's quite a horrific disease. So there's 3 types of NF, and I'm focusing on NF1 here for the sake of the presentation. And it's a tumor predisposition syndrome. So basically, you get huge benign tumors that can form in the Schwann cells of a human body. And they can either form cutaneous neurofibroma, which you see here in the picture on the lower side of things, and you can also have plexiform neurofibroma, which are these severe, huge fibromas which really impact your quality of life. So either way, I think you are horrifically affected by these issues, whether you want to call them cosmetic or functional effects. I think it's a horrific disease that causes a lot of social anxiety just because of the way it presents. But NF also has a role in the brain, so there's cognitive defects, and there's a risk of elevated stroke and bone abnormalities. And if you're so unlucky that you have neurofibroma, then 10% of these patients go on to have malignant brain tumors, and usually these patients die before they reach 20 years of life. So for a subpopulation, this is a really lethal disease. And at the moment, the disease isn't understood enough to understand who only has the kind of rather benign presentation of the disease to who ends up having the really malignant tumors, which is really impacting also the development of novel treatments for this disease. So that's the disease. And as I said, it's 3 times more prevalent than neurofibromatosis, but than cystic fibrosis, but it has a lot less approved drugs for it. So I think this presentation, there's now actually 2 approved drugs, but they basically do the same. They're inhibiting MEK, which is basically a repurposing of a proven cancer drug that's active in NF. And yeah, obviously, that's only basically treating the symptoms. That's not something that's curative by any means, and is actually associated with lots of side effects, which I'm going to talk through on the next slide, Laura. So the state of the art is actual surgery, and some of these patients end up having like 9 surgery within the years to remove these tumors, and they need to be constantly monitored. And there's a really, really high cost of healthcare for these patients. So I think the whole healthcare system spends 30 to 40,000 easily on average per NF patient. So there is a lot of money to play with in terms of health economics that is currently spent on actually looking after these patients. And then these MEK inhibitors that are currently out there, 30% of the patients do not respond from the get-go anyway, and 20 of them stop treatment because of the side effects. And yeah, I mean, MEK inhibitors have been developed for cancers and acute indications. They were never meant to be a lifelong therapy, which is actually what you would need in this case. And it becomes quite apparent why they might not help everybody if you kind of look at the disease pathway. So NF1 is actually an inhibitor of RAS that then triggers a lot of downstream pathways, and MEK inhibitors basically just hit one of those pathways. So there's a lot of room for improvement there. So that's the kind of state of the art. And basically the highest level outcome that you can potentially set is a curative one-time gene therapy that prevents all manifestations and comorbidities of NF. And I think in this case, we felt comfortable setting this kind of curative goal initially, because in NF, it's very, very clear what's the driver of the disease. So if we understand the driver, you can aim for a cure. So then basically we started with whittling down the requirements. So it needs to be something that overcomes this heterogeneity approach, right? It can't be something that's super expensive if people aren't actually super affected by it. There will be people who are a little bit more risk averse. So you need something that works with a health economics profile in a very diverse patient population. And you need to avoid toxicity, just because in this mixed population, there will be people who would not be up for something that's very, very toxic. And we're already seeing this with the MEK inhibitors. And interestingly, we thought this needs 100% uptake in the cells affected by lack of NF, because otherwise the cells that would not take up the new gene supplementation approach would always outcompete. So that's usually the case if you're aiming for a gene therapy. You want to have maybe 30% of cells transfected, and then you can argue that by achieving that rate, you will get the therapeutic effect that you're looking for. In this case, that's not the case. You really need 100% uptake, or at least we thought so. But then we also encountered something which I like to call false requirement, which gave us hope that something like that could actually be developed. So NF tumors are a lot less heterogeneous than usual tumors because they're all driven by the same tumor driver. So if you manage to hit it, you're actually getting a lot higher efficacy than with a normal tumor in normal cancer indications. And usually you think that for a gene therapy to work, you need to go in there prophylactically before these patients develop the disease. But here, just some really recent data had come out that just reintroduction of the wild type gene into established tumors actually kills off those tumors. So you could have the same therapy for prophylactic use and acute use, which then opens up a very interesting development path. So based on that, we homed in on NF1 gene therapy, and we thought that's definitely possible. We discarded some other areas that would have been really interesting to look at, and they were all basically triaged according to the areas of neglect, upside and downside, and then classified into kind of curative, disease modifying, enabling, tech, and preventative. But the NF Foundation, as well as we were, they're very, very driven to home in on that gene therapy approach just to see whether we can actually come up with something that's possible. So here's just some more thoughts about how we thought about gene therapies in NF. So NF1 and NF2, they are truly monogenic. So just replacing this one gene should have enormous effects. That's understood. There's a third kind of NF, which is called schwannomatosis, which is not understood how it would work. So it's not like a target for gene therapies, but the two NF1 and NF2 make excellent candidate. And we also homed in on that solution, homing in straight away on gene supplementation over editing of NF1 and NF2, because there are so many different mutations across these genes that there isn't a suitable market for a editing approaches. It's very unlike CF, where I think 90% have the same mutation that leads to the same phenotype. And a prophylactic treatment would be optimal because that could obviously prevent the development of many of the NF manifestations. But also, as I said earlier, you would have the same treatment that could have a curative effect in an acute treatment setting. And at that time, there was also some evidence that if you were to deliver NF1 genes in the academic setting, that you could regress and slow down the tumors. But none of the academic studies that we had actually seen were using vectors that could actually solve the challenges associated with the NF1 gene therapy. So how does it then look like? We homed in on our NF1 gene therapy, and then we figured out, okay, how would we be building an NF1 gene therapies? And then that's when you start designing constraints. So a constraint in this case would be the cargo capacity. NF1 gene is huge. So then, that excludes automatically usual suspects as AAV, for example, because they just can't fit that in. You need to have something that has specific cellular tropism because you want to deliver this to the Schwann cells. You also have another constraint, which is persistence in immunogenicity. You want something that hangs around a long time but doesn't cause any immune effects. And you want to have something with a proven safety record because of that increased need for safety that I alluded to earlier. So then we basically went through all the potential approaches that we could think of, looked at all the different vectors, and homed in very quickly on HSV, viral mimetics, and non-viral delivery vehicles. And then really whittled down is, okay, what would you actually be building, and how does a project looking at these three vectors turn out to understand which of them would be the optimal one? And then you end up triaging vectors, and then you end up with a huge table that looks very neat here, but I think took our founder in residence, who was working on this like 3 months to complete, to understand how all of that looks these days. This is a little bit easier with the help of our element tool. But basically, you create a table where you have all the vector features that you think you want to have, and then you basically map the different options against it. And you can see here that HSV comes out as a strong contender, as well as non-viral. Had a lot of question marks at that point, but we were really, really trying to understand this. And then in addition to all of these factors, we threw in other parameters that we thought would be very relevant, which would be the ease of manufacturing, the efficiency of delivery, and persistence in vivo. And yeah, so what came next once we had that table? Then we wanted to figure out which vector is best and how to get that vector into the swan cells. How do you go around the liver, and how do you tackle this 100% requirement? And at that point, we brought in a founding team. So our founder in residence, who is Liran, who's on the next slide, I believe, who had been driving the scoping to that stage. She is a serial entrepreneur, so this is her second startup that she's looking to start. She is an expert in gene therapy. She was working for Spark Therapeutics for 12 years. Was joined by Gonzalo. Again, he's a serial entrepreneur with expertise in gene therapies. So they both joined DSV as a team to want to spin out that company in the future and work out how to turn this initial idea into practice. And they worked together, I think, for 6 months before we incorporated the company. And they basically mapped out all the different questions that were still open. So we ended up on an approach that allowed LNPs to home to Schwann cells. So there was really good LNPs evidence that with some formulations, you could actually reach the Schwann cells, and that you could increase that with targeting to specific ligands. We are going after NF1 first because it's the bigger market and slightly easier than NF2, and have come up with specific systems that restrict the gene expression. And in order to get around this 100% requirement, we have come up with a tumor eliminator strategy. So this is not something I can talk about for IP reasons, but this is a molecule that has been in clinical trials with very positive phase 2 results, but needed something for efficacy. We'll need something that goes alongside a local delivery strategy, which is something that we have here. And around that solution, these guys then work through how we bring that actually to the market. So they've looked at various ways of administrating that, and we are going to go for a local administration strategy first, because that will be a much quicker route to market to then de-risk the overall value proposition. And that's a much quicker route to take than going for a systemic strategy first. Next slide. And even with that product, just for the leg, you end up with a $10 billion market. So a lot of people, when we initially spoke to them, were really, really worried. But just figuring based on our current assumptions of how much it will take to develop that therapy and sell it for, you'd still have a massive market that you could go after, which would increase if you further down the line figure out how to apply this systemically, which is kind of on the next slide. So that would be the strategy, how that company could grab even bigger markets. So you could go after systemic delivery, or you could pivot the company towards other Schwann cell specific diseases. And there's actually quite a number of them. And all of a sudden, you are left with a very interesting value proposition. And obviously, this project did not start with an IP starting point coming out of academia, but it developed the strategy to obtain IP along the way. So we factored in where the specific areas of novelty could be, and then started out the company with a solid experimental plan on how to obtain that IP, and that will be then protected in the first few months of that company. And yeah, that was a company which is now called Courage Therapeutics. It was spun out in 2024, December, so roughly 3 months ago. That team is now already in the lab, has the first experimental results already that have validated everything that we have come up with, at least in vitro. They are now bringing in philanthropic funding from the different funders in the area. They've already secured grant funding from further grant funding from the CTF Foundation. And they have good collaborations with leading research institutes, the Weizmann in Israel and the University of Barcelona, to tap into their local ecosystems where they're based. They have a patient scientific advisory board. They have lots of advisors from the industry that have some sort of link to the disease. And yeah, they have been going strong. And that was basically 18 months after we said, okay, how do we solve NF?
Great, thanks, Kerstin. So very quickly then, because I know we want to leave some time for questions, I just wanted to kind of loop back and tell you a bit about our broader track record, and then just explain how we work in partnership with other organisations, because the vast majority of our companies are built together with partners who share our desire to make an impact in particular therapeutic spaces. So I mentioned earlier that we work not just in therapeutics, but across a range of different sectors. So over the last 5 years or so, DSV has now built 49 companies across all of those sectors. Within pharma team, I think we are at 14. They're actually coming thick and fast at the minute, so that's why I hesitate a little bit to put my finger on the exact number, because we've been launching companies every couple of months for the last sort of 8 months or so. Start to finish, that process that Kerstin described takes historically around about 18 months. So that's going from a very, very wide problem that we wish to solve, such as Kerstin said, neurofibromatosis, or it may be some other significant large medical indication, and then right the way through to fully launching that company has taken us on average 18 months in the past. We anticipate that will actually speed up with the tools such as Elman that Kerstin mentioned, that just help us to accelerate a lot of the diligence that we do. The sort of start to finish cost of going from zero to company currently works out here at around this 450,000 mark. So that includes having those founders in residence on board, doing the scoping with us, all of the resource that it takes DSV to build the company, consultants that we might engage along the way. That's basically a kind of all-in cost of going from nothing on paper to a fully launched company, which I think, coming from the kind of grant funding world, is sort of within the realms of what individual translational grant awards might often look like, for example. Of all of these companies that we've built, 82% remain alive and kicking and undertaking work today, which for anyone who's familiar with spin out and venture statistics will know that that's very high. Perhaps counterintuitive, given that we start with, I guess, what we'd call a zero data biotech, with a sort of concept on paper and without their own data in hand. But I think what this number speaks to is the very sort of logical, systematic, almost engineering style approach that we use to go about building new concepts, where those individual components are de-risked in their own right, but perhaps have never been used combinatorially in the way that we envisage. And then also that the work plans that we put together are designed to, very early on in the life of the company, to prove that the concept has legs, and that allows the companies to go on and raise additional investment externally pretty rapidly in general. The current value of the portfolio that we've built so far is, this number's sort of ticking up all the time, but now approaching $500 million, which, considering the relatively modest cost that has been input into building those companies, actually reflects on paper a large return on investment. And the other thing the companies are really good at is then going on to secure not just venture funds, but also non-dilutive funds to help leverage against that initial investment. So on average, companies are securing then up to 24-fold the investment that was originally put in to launch the company. There's the names of some of our companies that we've built in the pharmaceutical sector over here. We've worked across a whole different range of indications, quite a number of cancer companies, because we've had a very productive relationship with Cancer Research UK, spinning out 4 companies now with them. But we've also worked across neurodegeneration, as Kirsten mentioned, neurofibromatosis. We have a cystic fibrosis company, and we have a number of companies in the enabling space as well, so unlocking bottlenecks within manufacturing, for example, like Plurify. And there are 3 others here that are not yet announced in stealth, so hopefully you'll see some news in the coming weeks and months about our next launches. Because of this engineering style approach, and I think something else that distinguishes these companies from classic academic spin-outs is they very often are combining knowledge and ideas from across what would traditionally be different silos. And I'm sure this is something that everyone on the call is familiar with in terms of trying to find mechanisms to encourage scientists to work with scientists of completely different disciplines. Taking this paper-based approach, then we naturally bring together information from across, for example, biology with computation and bioinformatics and engineering. So we often end up with companies in either an advanced therapy space or leveraging big data, or indeed combining those 2 discipline sets. I mentioned here that we work primarily in partnership with other organisations. I just put in some thoughts directly from our partners here that you can look through, when we share the slides after. But you can see on the right a really wide range of partners that we've already been working with to build companies across our sectors. But within pharma, you can see, for example, partners like AbbVie. General Inception is another venture studio that we've recently teamed up with. And then a whole range of non-profit partners here, in addition to the Children's Tumour Foundation. So we have projects with Cystic Fibrosis Foundation, with the Cell and Gene Therapy Catapult, with Innovate UK, amongst others. I just wanted to give you very briefly a couple of nuts and bolts of what partnerships with us actually look like. So obviously the most important thing for us is a strategic and cultural alignment with the partners that we choose to build companies with. So we need to share a passion for the opportunity area, for the the patient sector that we'd be looking to address. And ideally our partners are open-minded to working in this top-down way of identifying the need first and then engineering backwards towards the solution. So it's a leap of faith in a sense for some partners, because when we go into the collaboration, of course, we can't say, here's the specific technology that we're going to build, or here's what the team will look like. It really is a trust in the process and the track record that we will, in the end, arrive at a novel and exciting new solution that would not otherwise arise. So the partners tend to bring often a lot of disease knowledge, perhaps networks, and also then just the operational cost of venture creation, which is contributed, as I mentioned, often in a milestoned way. And then in exchange for that, the partner receives founding equity in the company and the opportunity also to support the company launch with equity investment. So yeah, just to wrap up then, in terms of how organisations can work with DSV going forward, we're always very interested to talk to potential strategic partners that we've not worked with before. We tend to have a bit of an internal wish list of disease areas that we'd love to tackle in our next projects, and I've put a few of them down here. But equally, perhaps in our DNA, we're also a very open-minded organisation. So if partners approach us with a compelling thesis for a problem to be solved, then we're very open also to considering that and deciding whether that's something that we can also get excited about working on. We also obviously work with organisations that can invest in early stage companies, and we work really hard to bring the right early stage investors around the companies we launch that can help to support them on their journey.